Hello friends, today we are going to see the fluid mosaic model of plasma membrane. We all know that the cell cytoplasm of animal cell is bounded by a limiting membrane called cell membrane or plasma membrane or plasma. The definition of plasma membrane is that it is the thin, elastic, semi-permeable living membrane that serves as a boundary for the cytoplasm. The term plasma membrane was given by C. Nageli and C. Kramer in 1855, while plasma was coined by J. O. Bloch. The size of plasma membrane is about 75 angstrom thick, but varies in different cells and ranges from 75 to 105 angstrom or up to 215 angstrom thick. Now let us see the chemical composition of plasma membrane in short. Plasma membrane is formed of mainly glycoproteins and phospholipids and small amount of carbohydrates, salts and water, etc. The lipid-protein ratio varies with the cell type. Example, plasma membrane of nerve cell has 79% lipid and 20% protein, while in mature RBC, the lipid-protein carbohydrate ratio is 40:52:8. These molecules are arranged in definite pattern. Models of plasma membrane. Various molecular models have been proposed to explain the structure of plasma membrane. According to some, it is in the form of lipid bilayer, while some shows that it is micellar. Let us see the bilayer models of plasma membrane. In this category, there are following models. First, the sandwich model, which is also called as protein-lipid model, proposed by Daniel and Davison. Second, the unit membrane model, which is proposed by Robertson. And the third, fluid mosaic model, proposed by Singer and Nicholson. As the fluid mosaic model is the most widely accepted model, we are going to see it in detail. Now let us see the fluid mosaic model in detail. The fluid mosaic model was proposed by S. J. Singer and G. Nicholson in 1972 and has been widely accepted. According to this model, plasma membranes are made up of double layers of lipids, mainly phospholipids, containing embedded proteins, forming a mosaic pattern. Hence the name fluid mosaic model. Some of the protein molecules penetrate through the lipid layer, partially or wholly. These are called as integrated or intrinsic proteins. These embedded proteins are important as facilitators in moving molecules through the membrane. The lipid molecules are organized in a bimolecular layer, meaning that the nonpolar region is organized in the middle, that is away from water as it is hydrophobic, and the polar regions are oriented towards the outside, that is the extracellular fluid and the cytosol. The intrinsic proteins allow the molecules to transport through the membrane while provide the barrier. According to this lipid model, the membrane pores are not stable. They appear and disappear. Fluid mosaic model explains asymmetry of membrane. Intrinsic and extrinsic proteins are not similar and do not form layers. Glycoproteins and glycolipids are found towards the outer surface only, which are antigenic in their property. Majority of enzymes are arranged on inner side, that is, on the cytosol face. Now this is the fluid mosaic model of plasma membrane. Here you can see the plasma membrane is made up of lipid bilayer and these are made up of phospholipids. The phospholipid is having a polar head and a non-polar tail region. The polar heads are hydrophilic in nature while the non-polar tails are hydrophobic in nature. Here you can see some of the proteins are embedded and passing through the plasma membrane and they are called as intrinsic proteins, while some are on the outer surface of the plasma membrane are called as extrinsic proteins. Here you can see the membrane pore as well as some of the sugar chains attached to the protein are called as glycoprotein, while when the sugar chains are directly attached to the lipid content, they are called as glycolipids. And these serve or give the antigenic property to the plasma membrane. Now, this is the 3D structure of fluid mosaic model of plasma membrane. Here you can see some of the proteins passing through which are called as integral or intrinsic proteins and some are the extrinsic proteins also called as peripheral proteins. When the sugar chains are attached to the protein, they are called as glycoproteins, while when the sugar chains are attached directly to the lipid, then it is called as glycolipid. And these are giving the antigenic property to the plasma membrane. And they are always lying towards the outer side. This model explains that membranes have several kinds of receptors. A receptor recognizes the chemical signals and carry out biological responses. The fluidity of membrane provides flexibility and suggests considerable freedom of movement to lipid and proteins within the bilayer, that is, flip-flop or lateral. Fluid mosaic model is the most accepted model because it explains the transport through the plasma membrane. Hope you understood the topic nicely. Thank you.